Welcome to the Deep Dive, the place where we don't just summarize complicated topics. We try to hand you the integrated roadmap so you can get straight to those essential concepts and the aha moments. Today, we are undertaking what many consider the Everest of biological sciences, immunology.
It truly is. This field is just immense, it's constantly evolving, and it can be, well, notoriously complex.
Oh, absolutely.
For anyone studying medicine, working in biotech, or, just a committed self-learner, the source material often feels overwhelming. It's so easy to get lost in the sheer number of molecules, cells, and all the acronyms.
And that's our mission today, really, to cut through that density. We want to provide an integrated high-level summary of the core concepts, focusing on the structure and the flow.
Right.
We need to understand not just what the immune system's parts are, but how they link up from the earliest defense mechanisms all the way to the most sophisticated clinical applications.
Exactly. We're going to structure this by starting with that foundational overview and then constantly showing you the connections, because immunology is, at its heart, this massive interconnected network. It's designed for two things: maximizing diversity and ensuring safety.
Okay, let's start with the fundamental division of labor then. The body's primary defense system is supported by two massive, distinct, yet cooperative pillars: the innate and the adaptive systems.
The innate immune system is your first responders. It's rapid, it's nonspecific, and it is always on call. This is the defense system you share with, well, virtually all multicellular organisms.
So this is the ancient foundational layer.
Precisely. It relies on physical barriers, but also on cells like phagocytes, neutrophils and macrophages, which literally engulf and destroy invaders.
And these innate cells, they don't need a specific blueprint to recognize danger, right?
That's right. They recognize these general molecular signatures that are shared by broad classes of pathogens, things like common bacterial wall components. They use generic tools like complement components, which can punch holes in microbial membranes, and chemical alarm bells, which we call interferons, to signal danger.
So a lot of the time, this response is enough.
Oh, often it's so effective it destroys the invaders before you even notice the fight began.
But when those general defenses are overwhelmed, or maybe the threat is totally new, that's when we call in the specialists, the adaptive immune system.
And this is the system that really revolutionized host defense. It's defined by its highly specific mechanisms and, crucially, by memory. It relies on lymphocytes, B cells, and T cells, and its strength is its ability to generate this just astonishingly diverse repertoire of receptors.
To anticipate almost any foreign structure it might encounter.
Exactly. It's proactive.
So if one is nonspecific and rapid, and the other is highly specific but slower to build, how do they communicate? How does the innate system tell the adaptive system, hey, we need you?
They absolutely have to communicate, and that connection is mediated by a vast family of chemical messengers called cytokines.
Okay.
Cytokines act as the immune system's localized command and control network. For example, interferons, which we mentioned in the innate system. They're actually a type of cytokine. They alert and activate the lymphocytes, effectively bridging that rapid, non-specific response with the slow-building, highly targeted adaptive one.
So the innate system basically hands the evidence of the invasion directly over to the adaptive system.
That's a perfect way to put it. And understanding this basic flow has enormous real-world consequences, even at the most fundamental level.
I can imagine.
It really does. When you look at public health, at infant mortality globally, our knowledge of the adaptive immune mechanism is arguably the most powerful tool we have. Simple, cost-effective measures like vaccination, which harness that power of adaptive memory, they dramatically reduce deaths from things like measles or waterborne bacterial disease.
So understanding the science translates almost instantly into life-saving clinical practice. That brings us to specificity. The key to the adaptive response isn't just the cells, it's the core recognition machinery they carry. Let's tackle the fundamental difference between B cell and T cell recognition, because that difference really defines their entire job description.
Let's start with the B cell. B cells are the engine of the humoral immune system, which means they fight threats floating outside our cells, extracellular pathogens. Their receptors, which are essentially membrane-bound antibodies, recognize the antigen directly, just floating free in the bloodstream or in tissues.
And the theoretical diversity is just mind-boggling. I think it's up to 10 to the 11th possible antibody variants in one person. That seems genetically impossible. How can our finite genome achieve near infinite diversity?
This is one of those aha moments in immunology. The system achieves this diversity by, Well, by essentially treating its own genome like a stack of building blocks and just shuffling them randomly. This is the generation of diversity through gene rearrangement or somatic
A
recombination.
genetic lottery, almost.
It is. Developing B cells randomly select and join these gene segments, we call them V, D, and J segments, and then use imprecise joining mechanisms to stick in extra nucleotides. This random combinatorial assembly means that nearly every B cell expresses a unique, previously unseen antigen receptor.
That contrasts that radical freedom of D cells with the T cell receptors, or TCRs. They mediate cell-mediated immunity, so they're focused on intracellular threats, like viruses or cancerous cells.
And the T cell operates under incredibly strict restrictions. The key distinction here is a concept called dual specificity.
Dual specificity, okay.
A TCR cannot recognize an antigen floating free in the body. It recognizes an antigen only when that antigen, now chopped up into a small peptide, is presented on the surface of another cell by a self molecule called the major histocompatibility complex, or MHC.
So I'd have to see two things at once, the peptide and the MHC molecule presenting it.
The peptide and the MHC. And unlike antibodies, the TCR is never secreted. It's strictly bound to the cell surface.
So the MHC molecules are the central presentation stage for all T cell action. How are these stages different, and who uses them?
We have two main classes. Think of MHC class I as the cell's internal status report. Since they are found on virtually all nucleated cells, they constantly sample and display what's happening inside. They present peptides to the CD8 plus T cells, which are the immune system's assassins.
The cytotoxic T cells.
That's them. In contrast, MHC class II molecules are chiefly restricted to professional antigen-presenting cells, or APCs, so macrophages, B cells, and dendritic cells.
These are the detectives.
The detectives, exactly. They pick up external evidence, they process it, and then they present it on class II to the CD4 plus T helper cells, who are the ones who direct the entire response.
That distinction, class I, showing internal status to assassins, class II showing external evidence to commanders, that really makes the entire system click. And unlike the randomly assembled receptors, MHC diversity is fixed, right? It's inherited.
Precisely. The enormous diversity, or polymorphism, of MHC is inherited. The MHC alleles you get from your parents are the alleles you have for life. There's no gene recombination happening here.
And this is why certain people are more susceptible or resistant to certain diseases.
Exactly. It's a huge factor in both infectious disease and autoimmunity.
The fact that the system generates billions of potential receptors and then must filter them, that leads us directly to the thymus. How does the immune system perform its most essential trick? Ensuring it attacks non-self, but leaves self completely untouched.
It does this through a profoundly brutal process called T-cell selection in the thymus. Over 98% of all developing T cells, they're called thymocytes, are culled and die.
Wait, 98% destruction? That feels incredibly wasteful. Is that just the immune system prioritizing safety above all else?
Absolutely. It is the cost of absolute safety. If a T cell escapes that checkpoint and attacks a self-tissue, the consequences are disastrous.
Well, how does it work?
The process has two steps. First, positive selection. A thymocyte must recognize self-MHC with medium affinity to survive. If it binds too weakly, it dies. This ensures the T cell can do its dual specificity job.
Okay.
Second, negative selection. Any cell that recognizes a self-peptide presented on self-MHC with high affinity is immediately eliminated via apoptosis. This establishes central self-tolerance and prevents widespread autoimmunity.
Okay, so assuming a T cell successfully navigates the thymus, it's mature but it's still naive. How does it transition from a surveillance patrol to a fully activated effector cell when it finally meets a non-self antigen out in the periphery?
Well, the first step is antigen processing, which depends entirely on where the threat is located. For extracellular antigen, say bacteria that an APC engulfed, the antigens are digested in vesicles and displayed on MHC class II for the CD4 plus T cells.
The helper cells.
Right. For intracellular antigens, like viral proteins, they are topped up in the cytoplasm, transported into the endoplasmic reticulum via a special transporter, and then displayed on MHC class I for the CD8 plus T cells.
And as we learned, simply seeing the antigen isn't enough to trigger activation.
Correct. This is our second layer of safety of the two signal requirements. Signal 1 is the actual recognition of the antigen MHC complex, but signal 2, cross-stimulation, is also required. This is usually delivered by a second set of interacting molecules like CD28 on the T cell binding to a danger signal like CD80 on the APC.
Why is this crucial redundancy built in? Why two signals?
Because if the T cell receives signal 1 but lacks that essential co-stimulation, that second yes signal, it enters a permanent state of dormancy known as anergy.
So it's shut down.
Permanently. This prevents T cells from accidentally reacting to a self-antigen that might be innocuously presented somewhere in the body. It guarantees T cells only become fully activated when there is bona fide danger, which is typically signaled by inflammation or infection.
Once they're fully activated, the CD4 plus T helper cells become the immune system's customizable commanders. They don't all follow the same playbook. They differentiate based on the threat.
Exactly. Different pathogens require different weapons. This specialization is governed by the initiating cytokines present during activation, and that determines the final T helper subset, or TH subset.
So what are the main types?
For threats like viruses or tuberculosis, which hide inside cells, the environment favors the Th1 response. Th1 cells produce interferon gamma, which is essential for activating those cytotoxic CD8 plus T cells and really enhancing macrophage killing power.
And for threats like, say, parasitic worms, the response profile changes completely.
That shifts to the Th2 response, driven by cytokines like IL-4 and IL-5. This response is tailored to fight surface pathogens and large parasites by promoting the production of IgE and recruiting specialized cells like mast cells and eosinophils.
The third major subset seems designed specifically for common microbial threats we face every day.
That would be the Th17 response. This is driven by innate signals when you encounter extracellular bacteria and fungi. Th17 cells secrete a cytokine called IL-17, and their primary function is to call in the cavalry specifically, attracting vast numbers of neutrophils to the site of infection.
Okay, now let's transition this massive knowledge base into clinical utility. Diagnostics rely entirely on the high specificity we've just detailed.
They do. Immunologic tests capitalize on the specificity and the high binding affinity of that antibody-antigen interaction. Take the ELISA test. This simple test lets us detect the presence of a specific antibody, say an antibody against syphilis, by using an enzyme-linked reagent to produce a color signal that's proportional to the amount of antibody bound.
And when we need to count specific cell populations, like monitoring T cells in an HIV patient, we use advanced technology to read their surface markers.
That is flow cytometry. By tagging cells with fluorescently coupled antibodies that are specific for surface markers, for instance, measuring the number of CD4 plus T cells, we can accurately count those populations in clinical samples. It gives us crucial information on immune status and disease progression.
Let's return to vaccination, which is really the ultimate utilization of adaptive memory. The hepatitis B vaccine is such a classic success story.
It's just elegant engineering. The vaccine isn't the live virus. It's a subunit vaccine made of recombinant hepatitis B surface antigen, or HBsAg. This protein naturally assembles into virus-like particles which mimic the external appearance of the virus.
So the immune system sees it, but there's no actual danger.
Exactly. When it's injected, the immune system generates high-affinity neutralizing antibodies against HBsAg. These antibodies, if you're later exposed to the real virus, physically block it from binding to the receptors on your liver cells, and that confers sterilizing immunity.
And what about the genius required to make vaccines against poorly immunogenic targets, like bacterial polysaccharides? That led to conjugate technology.
Right. The problem with polysaccharides is that they activate B cells but without T cell help. This leads to a weak, short-lived response, especially in infants. The solution in conjugate vaccines, like those against Haemophilus, was to chemically link the polysaccharide to a highly immunogenic protein carrier, like tetanus toxoid.
Do you trick the T cells into helping?
You do. Now, the T cell sees the protein component, gets activated, and provides that essential help for the B cell to generate a strong, specific, and long-lasting memory response against the polysaccharide.
Finally, what happens when this carefully constructed system, with all its checks and balances, selection, energy, co-stimulation, what happens when it turns toxic?
That leads us to hypersensitivity and autoimmunity. Hypersensitivity reactions are these overzealous immune responses that cause damage. We classify them based on their mechanism.
The classic immediate allergy is type I, right?
Type I, hypersensitivity, it's IgE mediated. It involves the cross-linking of IgE antibodies that are bound to mast cells, triggering this rapid release of powerful mediators like histamine. This is the instantaneous allergic fire alarm, the immediate swelling, the bronchoconstriction you see in conditions like anaphylaxis.
And type 3, which is damage caused by circulating debris.
Type 3, hypersensitivity. This involves circulating immune complexes. So, antigen bound to antibody. If these complexes aren't cleared efficiently, they can deposit in small blood vessel walls or filters like the kidney glomeruli. There, they activate complement and attract neutrophils, which cause tissue damage.
And this is what we see in diseases like lupus.
It's central to diseases like systemic lupus erythematosus or SLE and post-streptococcal issues.
So autoimmunity fundamentally is the ultimate failure of all those checks you discussed earlier.
It is the failure of T-cell tolerance. Autoimmune diseases, whether they're T-cell mediated like type 1 diabetes or antibody mediated like celiac disease or lupus, they happen when those central or peripheral tolerance mechanisms break down. And this almost always results from a complex synergy between genetic risk factors like specific HLA alleles and environmental triggers like infections.
That was a phenomenal synthesis. We've moved from the single lymphocyte and its random receptor assembly all the way to complex chronic inflammation and therapeutic interventions.
And the clinical translation is why this field is so critical. Every piece of core knowledge we discussed, from cytokine subsets to receptor structures, is being used today to develop treatments. Whether it's using anti-TNF monoclonal antibodies to manage rheumatoid arthritis or engineering stem cell therapies to cure devastating conditions like severe combined immunodeficiency.
For those of you who want to really cement this integrated understanding of different immune mechanisms of damage, let's leave you with a final challenge question.
Okay. Your task is to compare and contrast the destructive mechanisms in two distinct forms of antibody-mediated disease. Think about type 2 hypersensitivity, which involves IgG antibodies binding directly to a cell-bound antigen, for example, on a red blood cell in autoimmune hemolytic anemia.
Okay, so a fixed target.
A fixed target. Contrast that with type 3 hypersensitivity, which involves IgG-forming circulating soluble immune complexes, like we see in SLE. How does the site of complement activation and the resulting involvement of phagocytic cells, like macrophages, differ between these two processes? Specifically, where does the damage happen and why?
A perfect way to test your ability to connect the receptor to the disease gate. Thank you for joining us for this deep dive.